I'm now going to talk about field trip methods. Now, if you're looking at an area, it's impossible to count every single plant, every single animal. So instead, we take a sample, and if the sample is good, you get an accurate estimation of the species number or of the distribution of the species. So, say I want you to be able to describe how you would estimate abundance. And the first way you would do that is for plants, you could record the percentage cover of the plant. So you put a quadrat down and you would estimate the percentage of the quadrat that is filled or covered by that plant. That isn't that accurate, but it's certainly a very acceptable method. Another way to estimate abundance is to record the density or to work out the number per square metre. So you can do that using quadrats, and you can measure in the density. The number per square metre is really good for slow moving animals, but it's not really great for plants, as it's very hard to count the number of plants because it's very hard to see where one plant ends and another plant begins. So certainly it's a good idea, but it's hard for plants, but it's easy for slow moving animals. Another way to estimate abundance is to record the frequency with which plants or animals occur along a belt transect or along a line transect. So each species is just recorded as being present or absent in a quadrat on that belt transect. A pin frame records the frequency as well. Remember, that is a frame, and out of the frame these pins drop down, and if the frame is then moved along a line transect, and if the pins touch a species, it is recorded as being present, and if they don't touch a species, it is recorded as absent. So that again gives us frequency readings. So there are two types of sampling method. The first is random sampling and the second is systematic sampling. So random sampling, you use this when you're comparing two areas. You're not looking for a pattern, you're not seeing if there's a change in distribution. There is no change, like gradual change in the environment. So you're not looking to see how seaweed changes going down the shore. You're maybe comparing the seaweed in one shore to the seaweed in a completely different shore. Then you would use random sampling. Random sampling is good because it avoids bias. In random sampling, you need to have at least 30 quadrats. So that's a large sample size, so that will make your results more reliable. And if it is random, it ensures that the sample represents the whole area. So if you're asked why use random sampling, it avoids bias and it ensures the sample represents the whole area. So how do we do that? So this is the really, really clever queen, kick Charles right round. So you put you put two measuring tapes at right angles to each other, and then you generate random coordinates using a calculator. So that's really clever. And then you place the quadrats at the intersections of those random coordinates, and then you use a key to identify what is inside the quadrats. Then you record what you see inside that quadrat, and you just wouldn't do this once on a shore. You would set up a whole lot of those on a shore, and that would make your results a lot more reliable. So that is random sampling. The other type of sampling then, as you know, is systematic sampling. Please, whenever you're asked about this, remember when you use it, when there's an environmental gradient, when you're looking for a pattern. So when there's a change in the physical environment. So you might be wondering how the wildflower population changes from the middle of a wood to the edge of the wood. So there's an environmental gradient there, and you would use systematic sampling. So it's used when there's a change in the habitat that seems to be causing a change in the species that you find. So you sample along a line, basically in systematic sampling, and the line is called a transect. So the first type of systematic sampling is where you just have a single measuring tape and no quadrats whatsoever, and then you just move along the tape and you record the species that are touching the tape. Then you could set up a belt transect. That's what we did on the shore, and then you place quadrats all along the tape, and then you sample within those quadrats. If it's a very, very large area, for example, if you're sampling over a few kilometres, you would use interrupted belt transects, and all that means is you place quadrats at intervals along the tape, maybe every 10 metres, and that is good for a large area. And remember, you see at the end of your answers, always say you set up a number of transects, like repeat with 6 more transects, because that will make your results an awful lot more reliable. Now, you do need to know your sampling apparatus. So first of all, when you're sampling animals, you could use a sweep net, but that is used when you're trying to catch invertebrates, insects, things like that in tall grass. So you would move through the tall grass and sweep the net from side to side, and every 10 metres you stop and shake out what is in the net, and you would put it into an enamel tray and then record what's there and where you got it from. If you want to catch little insects that are just moving around inside the bushes, a good idea would be to use a putter. So remember, you suck on this end, and this end has a little bit of gauze to make sure that you don't suck up the invertebrates. And then this end is put over the invertebrate, and you bring the invertebrates back to the lab to identify. If you're sampling a very slow moving animal, like a limpet, think about the things we saw on the seashore, all the shells, they're slow moving animals. You would actually just use a quadrat because they're not going to run out of it. For little invertebrates that walk along the surface of the ground, you would use a pitfall trap. So you dig a hole, you put in a jar of some sort. It's really important that the carton or jar is level with the ground. Otherwise, the insects just wouldn't go into it. They wouldn't like climb up a hill to fall down in. And the other thing to remember is there should be a lid. A lid prevents two things. It prevents the invertebrates from drowning, and it also stops predators, birds, from eating the insects in that pitfall trap. Sometimes in questions you're asked how you would measure abiotic factors. So here are 3 good examples. If you were measuring the pH of soil, you would use a pH meter. If you were measuring the temperature of the soil or of water, you would use a soil or a water thermometer. Remember, every time you put the thermometer in, you should put it into the same depth, and you should let it sit long enough to equilibrate. That means give it time to come to the actual temperature of the soil around it. And another abiotic factor would be light, and light changes over the course of the day, so that can be measured with a light meter. But if you're comparing 2 different areas, you would actually have to take lots of readings at the same time of day to make those results reliable. So I mentioned abiotic factors. These are non-living factors, and I've mentioned these ones. They might also be called physical factors, and edaphic factors are soil factors. Now you have to be able to describe how you would measure the soil moisture content. So let me take you through this. You would collect a very small sample of soil, and you would put it into a crucible, and you would weigh and dry that in an oven. You would heat it to 105 degrees C. You would keep weighing it and drying it and weighing it and drying it till you have a constant mass, and only then do you know that all of the water has been removed. So to work out the percentage water in that soil, it's the mass of the wet soil in the crucible minus the mass of the dry soil in the crucible. So the only difference between those two is the water that has evaporated. And then you would divide that by the mass of the wet soil, and you multiply that by 100. So just as I mentioned, 105 degrees C there, that will evaporate the water, but it will not burn the humus. And then whenever you're bringing the soil to sample, bring it in plastic bags to stop the water evaporating from it. It might be sitting for a day before you get doing this experiment, so that would be a good little tip there. So then if you want to work out the humus in the soil, that's the organic content. It contains a lot of carbon. So you start off with dry soil in a crucible, and this time you burn off the humus using a Bunsen, and you keep reweighing it. Now when you burn humus, the carbon combines with the oxygen to give you carbon dioxide, which leaves, so that takes away mass. So you keep burning it and reweighing it until you get this constant mass, and only then do you know that all of the humus has been removed. So to work out the percentage humus, then it is the mass of the dry soil that you started with, and then you take away the final mass of the burnt soil, and you put that over the mass of the wet soil, and then you multiply by 100. Just a little tip or two about these soil practicals. If you're asked how would you improve precision or accuracy, you would say use a balance that measures to at least 2 decimal places. If you were asked about validity, that's not really accuracy, it's more validity. You need to transport the soil in a plastic bag. So that would ensure that water hasn't evaporated before you actually get to the lab. So that would be a more valid result. And then for reliability, you need to take a number of soil samples, not just one soil sample, and find out the humus and water. You would need to take a number of different soil samples.